Welcome to Stories from the NNI. I'm Lisa Friedersdorf, Director of the National Nanotechnology Coordination Office. Today it's my pleasure to welcome Aaron Lavick, Professor of Chemical, Biochemical, and Environmental Engineering and Associate Dean for Research and Faculty Development at the University of Maryland, Baltimore County. Aaron, thank you so much for joining us today. To get us started, can you just tell us a little bit about how you first got involved in nanotechnology?
Absolutely. So we started a long time ago. We've been at this for almost 20 years. And the work that we're most excited about really came out of some observations that I had done. I had worked in my PhD on spinal cord injury, and I had observed, as have many others, that when we have more bleeding in the injury models, we have worse outcomes. And so we wanted to try to come up with a way to stop the bleeding. And we were really surprised at how few technologies were out there to try to manage internal bleeding. There were different drugs, but they didn't work particularly well, and they tended to have a lot of off-target effects that impacted the central nervous system. So they weren't suitable for spinal cord injury. So my very first graduate student and I said, well, you know what, we'll build something. How hard could it be? We read the literature, we came up with a design, and we figured about 6 months to build it, and then we could test it in the spinal cord model. And 6 years later, we had developed a successful nanoparticle to help manage bleeding. And to this day, I still can't believe we thought it was going to be 6 months, but it shows you a lot about what we anticipated when we started the work.
I recall some research many years ago that was looking at electrospun fibers to develop a mesh that could be used in surgery, or first responders could use it, but it's something that you could just leave in. Now that's a different type of approach. Can you compare and contrast and talk about how mesh or gauze would play a role in how your particles work?
Absolutely. So you can imagine, being able to pack a wound with gauze to control bleeding makes a ton of sense when you're dealing with something you can get to. And, I'm old enough that I think about the TV show MASH, and invariably you saw their gut open and they could stuff stuff in it. And at least in the short term, that potentially buys you time. And that's a big part of what happens in trauma, is trying to buy people time. But if you're dealing with something like a brain injury or a spinal cord injury, you can't just pack something in there during the treatment. Maybe later on you can. And ironically, the work that we originally did was building a scaffold to help promote repair in the spinal cord. And so it was something you actually inserted. And that's where we saw, when we inserted it, we saw less bleeding, which led to this whole area of research. The idea behind these particles is you can inject them intravenously. So even before you know where the injury is or how extensive it is, hopefully you could administer these particles just like you would administer a blood product, and would help reduce the bleeding and stabilize the patient so you could get them to what we call definitive care.
So how do they work?
It's funny, it can be really simple and tremendously complex at the same time. They are little particles that are decorated with something called polyethylene glycol, which is a water-soluble polymer, and it basically creates what we call a water corona around the particles. And then at the ends, we have peptides. And these are small sequences of proteins that interact with the blood products. So in our case, we're interacting with activated platelets. When you have an injury, you have platelets which look like plates, and they become activated, and they look like stars. And at the end of the points of the stars, you have receptors that try to grab onto stuff. And in one of the receptors grabs onto something we call the RGD sequence. We're such simple people. We just stuck a bunch of RGD on the outside of these particles, and they act like bridges between the activated platelets. And by doing that, they really help to form a clot more quickly and stop the bleeding.
You mentioned PEG. Now, is that the same structure that's used in lipid nanoparticles?
Yep, and in the COVID vaccines, and I just want you to know I'm super excited because that's lots of data for us. Now, that's obviously given subconjunctivally or intramuscularly. You get an injection. It doesn't go directly into your bloodstream, but it's the same chemistry on the outside of the particles. PEG's used a ton. It's used in the pharmaceutical industry, it's used in the cosmetics industry, and it's used in the food industry. For a long time, it was blended into ice cream to help make it creamy and keep it from crystallizing. So we have a long track record with it.
You mentioned spinal cord injuries in particular, and when you have internal bleeding in a region around the spinal cord, you described how your nanoparticles work. Are there other strategies that you use in concert with these particles to improve patient outcomes?
Absolutely. And I just want to emphasize we're not in patients. We are still, in the very much the preclinical models, and there's been a lot of challenges with it. I like to say that if you were a rodent, we can save your life and reduce your trauma, which is great. But ultimately, there's a long path between that and humans that we're still working on. But yeah, one of the things about these nanoparticles is that we can load them with drugs. And that gets really exciting because you can imagine for something, one of the things we've tested is brain injury. And in brain injury, you have a lot of inflammation. And some of that inflammation can be good, but some of it can actually exacerbate the injury. So we loaded the particles with anti-inflammatories, and we saw that that actually reduced the extent of the brain injury, which is super exciting. We really don't have good treatments for traumatic brain injuries right now.
So I want to switch gears a little bit and talk about your research group. And you mentioned your first graduate student several years ago when you started designing the research protocols for the study. What is the makeup of your students? What does your lab look like?
It varies. Right now, I have a few graduate students. We're relatively small at the moment, and a lot of that has to do with COVID. We have sort of shrunk down. It is a really tough time to be a graduate student. So I have a couple graduate students. I have a few PhD students and a few master's students, and I have about 10 undergraduates. And I have to say, joining a lab as an undergraduate in the middle of a pandemic, it takes a certain amount of courage. And my students have been absolutely fantastic, really figuring out, how do we manage doing research and dealing with supply chain issues that are really affecting research and dealing with all the logistics about keeping each other safe and making sure we can still teach each other. We actually have a GoPro in the lab that my students will put on so they can show each other what reactions look like without having to be on top of each other.
Can you talk about the disciplines that the students are studying?
I have found that there is no one discipline that helps be successful in this. And honestly, the thing that really helps is sort of being game and being willing to do new things. I have had students from all the engineering disciplines, mechanical, electrical, materials, chemical, but I've also had students from biology and chemistry. And I had one student who was a photographer, and she really liked science. And she didn't end up doing a degree, but she worked with me for 5 years and is on several of our papers and was phenomenal in the research lab. So we have a really neat team of people that we work with.
So what type of research are the undergraduates able to do in your lab, and where do you see them go?
So it depends on the undergrad, and it depends on their time and their interest. And honestly, it's how much they're able to dedicate to learning. It's funny, it doesn't depend on the discipline. I had a student who, as a sophomore, reached out to me and said, can I join the lab? And he did. And we ended up, of course, going remote, I think 2 weeks later. And so he wrote a review paper. And I'm still completely and utterly humbled that he was able to write a really coherent, beautiful review paper. Most graduate students can struggle with that. And he did this. And then when we were able to come back to the lab, he came back and he runs his own experiments. I mean, he can make all the different kinds of particles and characterize them and really think in beautiful ways about how the different chemistries of the particles influence the biological outcomes. I've had undergraduates who are first authors on papers and who drive the science. I have an undergraduate right now who's the leader of one of our teams. And I've had undergraduates who participate and are part of it, and that's what they want to do. And so I really think it depends mostly on the eagerness and availability of time. It takes a lot of time to do this work.
Well, I think that it's great that you're giving so many students an opportunity to participate in a meaningful way. And do you have any advice that you share with your undergrads?
I probably share all sorts of advice. Whether it's good advice, I think, is the bigger question. I think the biggest thing I tell students is they have to do something they're passionate about. If they're working in my lab and they realize that this isn't their thing, that is totally fine. And then the question is, what is your thing and how do we help you find it? That doesn't happen a lot, I have to be honest. I'm lucky that they tend to come in and really like it. But the other thing, most of my students do go on to graduate school. And, some of them, one of my colleagues, now a professor at Hopkins, was one of my undergrads years ago. And she's amazing. I'm in awe of her work. And it really comes down to sort of talking about what they'd like to do and how we can help get them there. they're the ones that make the decisions that sort of drive the buses of their careers. It's just my job to try to support them as much as I can.
So, Erin, I just want to thank you for taking the time to talk with us today. This has been a lot of fun. Do you have any closing thoughts that you'd like to share with our listeners?
I guess the biggest thing I'd want to share is there really is a tremendous amount of brilliant science going on. And I think about how many of my colleagues are doing extraordinary work while taking care of their families and trying to juggle things none of us ever expected. And I know they are very tired. And they keep going because they love this work so much and they love the potential of helping people. And I guess I just want to acknowledge how extraordinary that really is.